previous student, and it was so interesting what she was talking about. So it was so exciting and thinking, oh my God, this is a very interesting audience with a lot of insights over there. So I'm going to introduce myself a little bit so you know where I'm coming from. I'm actually a chemist, but I work in the pharmaceutical industry for a long time. And then I have this lecture done in 2 parts, just in case there's maybe questions in the middle and we go to the second part. So this is me. So I'm from Spain, so I have an accent, so proud of that. So I'm sure, you hear that. So I always say to my students, feel proud of yourself, where you're coming from, your origin, your parents, your family. Diversity is a beautiful thing we have, So I am a chemist. I did my PhD in Spain in natural products. And I went to another city. I did it in Malaga, in Madrid, to do a postdoc in natural products. And NMR spectroscopy has been the technology that I've been working for many years, And then I went to the United States. I always wanted to explore how is the life in the different country, what experience I can have. So I continue working in natural products. And then after that, I met my husband and I decided to change my life. And I say, okay, I'm going to stay here. It was not part of my plan. So I found in San Diego, because he was in San Diego, a postdoc in something that I'm not really much. I'm a small molecule person, and I'll tell you what it is in few slides. So I work in a protein DNA complex by NMR, whatever. But then I have to look for a job. Always we have to look for those jobs to do our lives and our professional development. So temporarily, I found a position in, I'm going to put the pointer if I can, so because sometimes I might use it. So I found a position as assistant professor of chemistry in San Diego. I taught organic chemistry and lectures and labs. This is more the area that I had more expertise. And then again, it was for 1 year. So looking for a job, I was thinking, where do I go? Industry or academia? So I applied for everything. And I ended up landing in industry, in the pharmaceutical industry. I've been there for over 25 years. So I worked for Merck in the East Coast, in Rahway, New Jersey, for 5.5 years in the drug metabolism and pharmacokinetics department. So I started really living the pharmaceutical industry from a scientific standpoint. I'm not a business person, I'm a scientist. And then I wanted to explore more areas, and I moved to Amgen 20 years ago. I don't know what happened. Time flies. My kids were very small, and now they are just young adults. So I've been working in the process development, another part of the pharmaceutical industry, and you will see this a little bit later. And I am currently a senior principal scientist. I have a group of NMR, this technique. This is a similar instrument like the MRIs for the hospitals, but I use this instrument for determining the structure of compounds. Do we have the right drug, the right impurity? Can we determine the structure of impurities for the process of things like that? So that's kind of my specific job in industry. But anyway, so because I'm a pure scientist and I'm not a business person, but working on an industry, I wanted to understand, at least in a personal view, business. So at the time I was having young kids, so I took online classes, not to really change my career, but to kind of be better integrated on my industry and understanding. So and then since 2014, as I kind of enjoy, you are the young generation, the future. So I think us, like a Professor Kroll and me, are the links for your future. So since 2014, I've been part-time lecturer for the chemistry department. And since 2016, I've been teaching drug discovery and development science, but then with another professor teaching the business. Now I'm doing both in general, as I said, for online. And I kind of enjoy, because I'm in this industry for more than 25 years, which is my daughter's age. So I'm going to talk first about more on the definition, what is tech transfer, and then more details. My second part of the lecture is going to be more details of what we do in the pharmaceutical industry. So the first thing I want to do is a simple definition of tech transfer. Who does tech transfer? Why do we do it? And the typical methods, and what is drug discovery and development phases, because where do we put this transfer there? Why do we need it there? So that's what I kind of towards the end. So what is tech transfer? So this is just a simple definition. So tech transfer is a transfer of a technology, a process, that we make as a discoverers to to a third party or a second party, for most of the time is whether for a scale-up production or for commercial manufacturing. So, and I'll explain why we do that. So in the pharma industry, what do we need to transfer? What requires to have tech transfer? We really need to know very well our products, our processes, our equipment, our methods. I'm focusing more on transferring how to make the drug substance or the drug product, the medicine, but we also transfer technologies that are used to determine how our processes are going. Now, one important part of the tech transfer is documentation. Everything has to be documented. Every single thing, the good and the bad, every single thing. So that is really huge. And also it's very important that the two parties, the developer and the partner that is going to do the process on the scale up, needs to have the right skills. Otherwise we fail. That's very important. So who conduct tech transfer? The typical way to conduct tech transfer, we go from government labs to private sectors. We can do tech transfer between private sectors in the same country or in different countries. We can also do from academia to private sector, and also a lot of collaborations with academia, government, and industry. An example in the non-industry, the non-pharmaceutical industry, is the GPS. The GPS was basically used in the military years ago, and on that, and it was transferred to us in the regular people. We use GPS on our phones to drive to places, those kind of things. This is a typical tech transfer technology thing, but we're going to focus on the pharmaceutical. So why do we do tech transfer? So these are three main reasons, but there's more. So first of all, the first reason I have here is lack of manufacturing facility. Okay, so the developer might be very good developing the technology, the product, but then it might be for smaller scales. But when you have to do large scale testing, the developer might not have the right facilities, the right equipment. So they will have to partner with a second party to be able to do that. We will do the tech transfer. That's one case of scaling up manufacturing. When I say manufacturing, means not just commercial. It means also maybe during clinical phases where we need to produce more amount of the drug to be able to have enough for the supply. Another case of reason of tech transfer is lack of resources to launch a product. So the originator might be able to conduct and produce the drug for phase 2 and phase 1 clinical trials, but when it becomes phase 3, large amounts, or become commercial, that requires large manufacturing facilities. So that's another way to find another organization that can help you on that. So you have to transfer your technology to them. Another one is lack of marketing, distribution, and distribution capacity. We're talking about production, but not only production. Let's say, yeah, we can produce the drug, we can get it approved, but how do we make the drug knowledgeable to our clients, which could be insurance company, hospitals, patients, other healthcare organizations? That's a difficult thing. So marketing departments are very specialized in areas that the company might not have that expertise. So tech transfer is not only just the technology to make something, it's also how to distribute it. So that's another example, So common method of tech transfer, and I'm not an expert on this. Professor Kroll is the expert, I'm not, but this is what I cannot teach in general my students. So this licensing. So we give another party permissions of our intellectual property to use it, for the convenience, for this, whatever we need to do. So the in-license strategy would be whoever obtains that permission, to use it with the intellectual property things. So, for example, the large company that is going to do the scale up of our drug for whether clinical trials or commercial will be that in-license strategy. That's whoever. The out-license will be the small molecule that give the permission to the other party to be able to do what they need. So will be the small company giving those intellectual property. They will get revenues. They will get other things, revenues. Forgot the name. Anyway, so payments for using those things and to scale up. So here is a simplistic roadmap of the drug discovery and development phases. We have the drug discovery first, in the pharmaceutical industry, what we have something called leap optimization and preclinical. We're looking for, let's say, we want to, our focus is maybe a drug in diabetes, right? So we want to look for a drug that is going to lower the hemoglobin A1C to make this and be able to have insulin release, whatever, to the patients. So we're looking for, we do a lot of testing with many drugs, testing them in vitro and in the discovery to find leads. And then we also do optimizations, maybe chemical derivatizations that make it more active to find the final candidate that is the best. We call it drug candidate. When we have all this information from the animal testing and in vitro testing and all the things, we have to write a big document, which is an application, IND or investigational new drug application, to request from the regulatory agencies like the FDA from the US or any other countries from Europe, from other places, testing in humans. Anything that has to be tested in humans is very highly regulated. Nothing can go wrong. I mean, we try our best. So then we go into these phases, phase 1, phase 2, phase 3. I have a slide, and the next one explain a little bit more in details. So phase 1, the first time we expose to humans this drug is called FIH, also first in humans. Phase 2, we prove the concept. Is the drug doing the right job? Is the drug doing the lowering the hemoglobin A1C for the diabetic people or whatever, And the third one is a large scale confirmatory, because the phase 2 is a smaller scale. So can we really make sure that this is also effective in other patients? When we get all this information and it's really good, and it's demonstrate our goals, then we have to file again in the case of the FDA, will new drug application or other regulatory agencies to look for approval for commercial. And then we launch it, but we still do more phase 4 clinical trials, because once we expose the drug to the whole world, right, or the whole country, whatever, we get approval, we still have to look into potential side effects, other maybe off-label, make sure that the drug is not off-label use, and maybe extend their application. There's other things. So in the next slide has a little bit more details. So we do the preclinical. Basically, we test tons of compounds to find the best one that give us in an in vitro and in animal testing, assess safety and biological activity. When we get that information and we file to IND for the FDA or any other regulatory agency, the phase 1 is only for a small group of healthy volunteers. They don't have the disease. Then we still do more animal testing, but we want to make sure that this drug is safe. It doesn't do anything wrong in people, right? And we test some single doses and multiple doses in a range that we've been predicting that is a good range for humans. And then, so if everything goes well, we go to phase 2. We test patients in the hundreds with certain percentage of the disease, certain amount of disease, to see if it does the job for efficacy. Obviously, safety is always tested, and we still continue testing different doses because we want to narrow down maybe 1 or 2 doses for commercial purposes, the best ones. And we also test with some interaction with food, some other drug. We start doing those kind of things and start looking at the side effects. When things goes well, we get to phase 3. We test thousands of patients, many times different countries, patients with different stages of the disease, and large populations, different ethnicities, different genders, different ages, multi-site trials, different places. We need to confirm the efficacy, and because the phase 2 was a small trial. You can have a beautiful small trial, everything is great, and you can expand it, and then the percentage, the statistics don't reproduce the same. Then you need to start looking about what was happening. Why this, so if it goes well, you see, it costs way more money than this. This was an estimate a few years ago. Now, this is not just one drug. You see here, 8 drugs enter the trial. We don't do 8 drugs eight drugs is an example. We don't do several drugs at the same time because it's a very expensive process. We go with the drug that initially we got as a first one doing well. If it fails, we still do research to get a second one. So we go back and forth until we get the one that is going to go through all the 3 phases and potentially giving us good results. When you give us good results, we make this big document NDA or new drug application to the FDA or any other regulatory agencies to look for approval with all the information collected from preclinical and clinical phases. And then once there is a time of approval, and when there is approved, we continue doing testing, because now the drug is in the whole world exposed, or maybe whatever country has been approved, more type of patients. We want to make sure we can have understanding of potential side effects that they didn't show during the clinical testing. And we also want to look into maybe expanding the label if there was other benefits for other situations or other diseases, especially like a cancer or things like that. We look into other cancer or something. So we continue doing clinical testing when the commercial drug is out there. This is much more detailed. I'm not going to go into that, but just to tell you, phase 1 is actually phase 1A and 1B. Phase 2 is actually 1A, 2A and 2B. Phase 3 and the phase 4. And for cancer drugs, we normally do phase 1, and phase 2. If it looks very promising with good results, it might get preliminary approval. And we do phase 3 when it's already commercial. The reason for that is there is certain cancers that they're very deadly, and there is something that could prolong the life of patients and provide some quality of life. So the FDA, understanding that, or other regulatory agencies, we can seek approval a little bit early, only for those cases. And when we do phase 1, just to make a note, we don't use healthy volunteers because cancer drugs are very toxic. We use cancer patients from the beginning and compare with the standard care, maybe chemotherapy or something, to see if this drug gets or a combo gets better. So we have a different group of patients with different drugs to compare with other drugs in the market to see if the new drug has any benefits or effects, so things like that. And normally I'm focusing in 2 modalities, which is the small molecules, the traditional pharma, for many, many years or centuries, and the biologics, which is the more new biotechnology kind of industry. So there are other modalities like gene therapy, or things like that, but this is the major ones, so there's more drugs in the market. So the small molecules are very well defined in their structure. I don't know your knowledge on chemistry, but this is an example of a molecule from one of the preliminary statins approved in the market for lowering cholesterol. And the large molecule example, this one is from Amgen. Actually, the 2 companies I work at, I didn't do it on purpose. I don't know. So this is Neupogen. This is a very complex protein molecule that stimulates the production of white blood cells in the body, and is used as a side effect for chemotherapy, because the chemotherapy drugs, they're very, they basically wipe out your immune system, and the patients are very sensitive to infections. So that's used to compensate side effects. So anyway, you can see, so the structure of a small molecule is very simple, can be easily determined for standard spectroscopy techniques, which the structure of the protein is quite more complicated, very sensitive to the 3-dimensional space, and not so has too many thousands of atoms, more difficult to define. The small molecules, normally they are produced by synthetic chemical synthesis, sometimes isolation from natural products, but we normally end up doing synthesis. And these are, not easy steps to do, but way easier to control than how we make the large molecules, the biologics, because they're made by the cells. We have to put on cells the right clone with the right gene to express that protein. And cells are not robots. They are very sensitive to pH, to temperature, to conditions, and they could potentially produce not just this molecule, also some derivatives. So we have to control very highly how they produce it. It's much more difficult to control. So a small molecule drugs, normally they are distributed for the patients in tablets or capsules. It's easy for patient compliance. There are some that could be a solution or could be an injectable, but majority of them, they are like an oral taken by mouth. And they are relatively resistant to the acidic of the stomach, so it can go by mouth easy. Then biologics and proteins, so those they break in drugs are easy to produce. They're basically copycats of the small molecules. They can, easily can reproduce the synthetic process, even if they do with different raw materials, but they can get to the right product. These ones the biologics are more difficult because they're more sensitive. How the companies doing biologics, the biosimilars, which is the generic of biologics, they have to be very high expertise on biology, on cells, on all those things. So it's actually more restricted on the type of companies that they can do this, because this is like a soup. Not 100% the cells will produce exactly the same molecule, maybe 80-90%. There's always a 5-10% that there's some modification. So we also have to look into those additionals. They are not harmful to the body. So that kind of a general difference on the two type of modalities in the pharmaceutical. Here is how we produce in the scale-up manufacturing the small molecules. We buy the raw materials, so we get them from different sources. We go to these manufacturing facilities to do the synthesis in big, large reactors, and we produce the drug. We call it API, which is active pharmaceutical ingredients. But this is not what the patient gets. The patient gets the drug in a form of tablets, capsules. It's mixed with excipients. These excipients are chemicals that helps potentially, like for example, maybe you have heard that you take a tablet to have a slow release. So it's convenient that the drug is released slowly in the body to reach the target for whatever reason or something. Sometimes it's for stability reasons or for protecting from the stomach. So we mix with the excipients, and this is what the patient gets. But the patient doesn't get it like that. We have to put it in a primary packaging, and then in a secondary packaging, normally a box or something. Now this might look like it's silly, but this is very important too, because there are drugs that are sensitive to light, or there are drugs that are sensitive to humidity, or people that live in the North Pole versus the equator, they're going to have different temperatures. So we have to look into compatibilities with the materials of the packaging that is not going to affect negatively to the tablets or something. There's a lot of studies here also. So for the biologic, we go to the cells. We have a group of cells, the master cells, the cell bank, that we know they are good for that particular biologic that we target. And we get a group of those. We always have to have sufficient for the future, for the next 200 years, So this is a cell bank. We take few of those, we cultivate it, we grow them, we put them, the clone has the gene that is going to express the protein or the biologic of interest. We produce that, we express the protein, and then we have to formulate it. We cannot just inject it in the body of the human, because we have to have the right buffer that is compatible with our body, with all, so that's what is the formulation. And then we have to put it in sterilized bottles or syringes, lyophilize it if we do it on solid, or it's on liquid, and then it gets packaged to the patients. Those packages have similar studies. We want to make sure that our drugging solution is not going to be degraded depending on the composition of the bottle, of the syringe. There's a lot of studies for even patients' compliance, not even patients' complaints, to see if, we need to make sure that these solutions are stable, particles don't form or things like that. There's a lot of studies also on the packaging for the biologics too. So here is again the drug discovery and development roadmap. So normally the area we do more tech transfer is actually in the phase 2 and phase 3, where we maybe transfer the method to a company to do the drug substance in large scales for us, and maybe to another company to formulate it with the drug product. And we have to also transfer the analytical testing. Sometimes we don't know if our drug is going to make it to the market, so we decided to use our facilities for other things, and kind of rent facilities from other partners to do this tech transfer and develop that. But it depends on the interest of the company. We also do tech transfer here, depending on the company facilities, but many drugs that may be produced in the market can be done in a different country. Antibiotics, actually, it's done, most of the antibiotics, even the new ones, are done in India and China. instead of in the US. This has been a bit controversial, especially with the problems with supply chains we've been dealing, and especially with the pandemic. But anyway, so a lot of the tech transfer in the drug development happens in the middle of this phase 2 and phase 3. This is just a glossary, and this is the first part of the lecture. If there is any specific question, or I can move on to the next part.
I had a question, real quick.
Yes.
For tech transfer, if it's like within the same company, if like you're transferring a process, say from like one site to another site, is that still called tech transfer? Or is there another terminology for it?
Actually, we call it tech transfer, even though we don't have to do any transfer of intellectual property because it's the same company. For example, we do research and development in a place in the research development part. Like for example, here I am Thousand Oaks at Amgen. But then we have a manufacturing facility in Rhode Island that they need to produce the drug, whether it's for clinical testing or whether it's for approval. They don't know how we make the drug. So we have to transfer all the processes. So it's the same thing. It's just internal tech transfer. So that's a good question. So I've been talking more about external organization, but we also do tech transfer ourselves, but we don't have to deal with the issues of intellectual property because it's internally on the company. But we have a big manufacturing facility and they need to know everything to make sure they could produce the drug at the same quality and the same test done and the same thing. So yes, that's a good question.
Thank you.
Hi, I also have another question.
Yes.
You said you work in process development, right? Do you happen to
Yes.
work in building 30?
Yeah.
I do too. I work in the pilot plant, and so does Roggy. I work on the
same
upstream
here.
team.
Well, I have an instrument on the basement in building 30 and another instrument in building 29. Even though I work in the small molecules, but we have an instrument over there. So yeah.
Oh,
So
small world.
Yeah, I know. So any other questions? So maybe I move to the next part. So I think I'm going to move to the next part and we can have more questions at the end. I don't want to take too much time of your class. I think your class is super interesting. So anyway, so the next part is a little bit more specifics. So what are we transferring? So, but looking into the technology transfer roadmap, the process development workflow in the pharmaceutical industry, the phases of the technology transfer, the flow charts, the technical package for the drug substance and the drug product, what do we say there, right? And the key drivers for success and the failures, because we also have failures. And some examples of CMOs, so contract manufacturing organization. So this is kind of the roadmap. So there is a research and development process where we do a lot of research and development, and we might create something that become an invention. If it has the right characteristic of something new, something never developed before, whatever. This invention will be assessed for technology if this is something that could be considered for intellectual property. It matches the characteristic of intellectual property rights, and then this will be something that we want to expand it and we want to use it. So we will look into technology marketing, especially we want to have partners to scale up or do some of this job. We will do the licensing part and then create some products and services, and we will get some revenues. We will get money and we can start again the process. We can get another invention or things like that. This is kind of the kind of a roadmap. So the workflow means we will have a research phase, a development phase, and a production phase of this technology, of this product, So the research phase is also looking into the quality and in the small scale. Then when we go to the development phase, we're going to look into a factory production, larger production. We need to maintain consistency on the quality of this product, specifications. We need to understand what those are, and also consistency on the development and the manufacturing, at the different batches, at the different scales. So we do this transfer from the research to the production, or from the development and from the product to the production. So in the production scale of phase, we also have to look at validation. All our test methods need to be reproducing the values. I mean, need to be reliable. We need to, we do a lot of validation testing and a lot of documentation about those steps of production. And then we are thinking about technology transfer to marketing the products also, you know, not just producing in the large scale. We want to get feedback on the production so we know how to expand to the marketing of the products. So here is what we do on the phases of the tech transfer. The first thing is all the documentation needs to be ready for, and I'll show you a list of information we normally do for drug substance and drug products, you know, on the production of this new product or new technology. We also need to know what kind of method, analytical phase, the testing method we are going to be using, that our partners need to be ready to use them. Then we do, with our partner, we do a testing in a small scale, is the pilot phase. I mean, we test ourselves also, you know, to make sure we can transfer, right? So we do a small batches where we test all the analytical methods on all the processes to produce our drug, whether it's biologics, whether it's small molecules. We have to do a large batch production to make sure that when we scale up, this is, you know, transferable, you know, and we do this in the manufacturing facility of our partner to make sure they can do it. Then we do to the manufacturing production batch. This is the real one that we're going to use for our testing, or whatever we're going to do for our filing, or things like that. So there is a lot of process from one after the other to make sure we can go from the small scale to the medium to the large scale, and it's reproducible, it's validated. Anything that goes wrong, we have to investigate it, you know, and remediate it. So there is a lot of things we have to look into. This is kind of a flow chart. We have to look into the quality assurance, the manufacturing productions, the quality control department, the packing production, engineering department, packing development, analytical development, and formulation development. In the next couple of slides, I just define what those things mean, which is part of the tech transfer. So the quality assurance is basically the guidelines for all the operations and all the processes that we're going to use, you know, to need to do to produce our product. All this documentation needs to tell us, you know, to be able to produce it from the beginning to the end. The manufacturing productions, a lot of documentation on production of this, from the materials used, the equipment used, the procedures, you know, instructions to get to this final drug. The quality control, all the analytical testing done in the starting materials, intermediates, drug product, you know, all the to make sure that we have control on all this equipment, you know, all the analytical testing needed to meet the specification of high quality of the drug. The packing production, as I say, packing is also important. We need documentation on how to produce those packing methods, those equipment needed, and those materials, you know. So engineering department, when we go to scale up manufacturing, we need engineers and documentation of all this equipment, you know, facility that can support this complicated equipment for scale, and these reactors, these other things, other typical other equipments, dry the product, whatever. Packing development, documentation for the testing. You know, one thing is making the packing things, and another thing is testing that this packing is not affecting our drug. What is the storage conditions? What is the stability testing? Because sometimes we've been seeing that there is unleachable components going to the drug and they can affect the quality of the drug. So we have to make sure that there's no interaction between this glass from the bottle or whatever is or the blisters from that is packaging our pills, or the syringes. You know, there is components in the syringes that has some oil for the barrel of the syringe. We need to make sure this doesn't go to our drug. This, you know, there's a lot of things to look into that. Analytical development, all the documentation, and also the equipment we're going to use, because we need to make sure that our partners are going to be able to reproduce the same equipment, the same testing, you know, very, very important. And to have to control the quality of the materials and the intermediates and the final product. And the formulation department, documentation, production, the equipment used for all those formulated things, for the drug product, which is the drug with the excipients. So now, what do we normally have? What kind of content we have? The drug substance is the drug itself. We'll call it API, active pharmaceutical ingredient. We need to provide all kind of information in this tech transfer. We need to provide what materials we're using, what vendors, what the specifications. We need to provide the process development history that we all the batches we have been doing. We need to provide that information to our partners so they see how we did the process. If we encounter any problem, how we solve those problems. These IPCs, which is in process control, to control from the research and development, so to the scale up stages, so we know how we control the quality of the material. How do we storage these things? How some materials are not stable, or it requires refrigerator, or requires control of the humidity. There's many things that can happen. Mass balance. We make sure that what we produce from the beginning to the end, we're not losing material in the middle, or why it's happening. Safety and environmental things, very important, because we need to control the environment. We need to make sure we know what to do with the waste. We produce a lot of waste when we scale up production. Analytical requirements, all methods, protocols, validation, everything basically it's a very extensive list. We need to make sure our partners has all this information so they know exactly everything, how to do it, and what to expect. For the drug product, which is the API or the drug plus the excipients, we also need to provide, where do we buy the excipients? Where is the grade? Where is the suppliers? Where is the quality? And basically same information, the batch manufacturing history, the specifications, how do we control the process control, the manufacturing records, the storage of the materials, all the reports as we have been going through the process internally, the stability, cleaning procedures. That's a big thing, because when we create, let's say we use a reactor and we create this drug or this step intermediate, and we have to clean this for the next time we use it, whether we use it for the same drug or the different drug. Any contaminant coming from using equipment that we use for other things is a huge no-no, a big problem, a red flag, So all this information needs to be listed. So what are the key drivers of success? We have quality, technical, and business. Quality of our products that comply with our specifications, and anything that goes out of control, we have to open an out of a specification investigation and document it. Technical, all the scale up, transfers, change, we have to do any remediations that could potentially, when we scale up, things might not reproduce exactly what it was in the smaller scales, anything, and optimizations, anything needs to be added to make sure we're going to be successful. And on the business, we have the timeline pressure, how long it's going to take to produce the product with the right quality, and how much it's going to cost to run the plant, all the equipment, the people, everything. So these are big components, drivers to see if the process is successful. But we also have problems and things fail. So examples of failing here, some inconsistency on communications between the donor and us. So we need to make sure that some problems could come from the raw materials, not the right quality, the equipment, the discipline, the people, education that can do the job. Many factors can happen that could not go well if our partners don't have all the things that we need, Failure to scale, they have to make sure that they have the right equipment to scale to the scale that we want. And instead of try to use a small equipment to try to produce the large batch, that's not going to work. Also need experience management. It's very important because we need to, this is complex process. We need people with expertise. If we don't have the right people with expertise, we're going to have a failure already. And a lot of showstoppers here is, absence of clarity on responsibilities, lack of communication with the partners, problems with the analytical methods, that they might not have the same equipment that we have. We experience this a lot of times, and then they cannot reproduce the same method, and then they see something different that we've told them, you have to use this method with this machine, and this is how you analyze the quality, and they use different method, and they're not going to be able to know, well, if we're seeing the same, things like that. If the documentation is inaccurate, starting from us, that we're going to transfer, that's going to be a problem for sure. Failure to assess issues, standard procedures not clearly defined, and improper planner. Here is an example of a study done by these researchers here, Devil Pali, published in the World Journal of Pharmacy and Pharmaceutical Sciences. They studied 40 companies, what is called contract manufacture, that they do outsourcing in tech. They are using tech transfer, right? So of these 40 companies, they observed problems. 60% of the problems came from quality control. The quality control of the products were not the desired one that they're supposed to produce. Flexibility on the production, 50%. Loss of intellectual property, this is very scary, 70%. Problems with management. Let's say you deal with a manager from the other company, the partner, and they understand your process, you discuss all those things, and then this person leaves the company, a new person comes back and doesn't know the whole thing. You have to start from scratch again, communicating all that information. That is a complete delay, and it's a problem. Time delays is a very big issue also. 60% of those companies couldn't comply with the goals to have the product to deliver. Let's say you have to do clinical trials, and you have all your hospitals, doctors, clinics ready. They're supposed to receive the drug next month. And you have this partner that is producing the drug to develop, to distribute it, and they're late. You spend a lot of thousands, millions of dollars. You have to delay everything with hospitals and everything to do these clinical trials. That's money cost, very, very high cost. So there are a lot of problems, and that's why you have to really do a good research for your partners. Make sure that your partners that you're going to transfer the technology, you're going to do what you're expecting to do. So we learn a lot of lessons by many failures, This is just examples, screenshots of companies that they not only produce their own drug, they also do tech transfer for others. Even Amgen, that sometimes serves as a tech transfer company, because we produce the drug from another company because we have facilities and trained people, things like that. So even big companies, they also do those things. We collaborate with others to produce their products, And here is an example in the 90s. Ranbaxy was an interesting company. It doesn't exist anymore. It was a very big company in India to do development or scale up in collaboration with others. For example, in the 90s, they collaborated, they contract, collaboration with Eli Lilly to develop a cost-effective process for this antibiotic, cefaclor. They also work with Bayer to develop a formulation molecule for ciprofloxacin and another antibiotic. But this is an interesting company. There is a book called The Bottles of Lies. It was published, I think, in the beginning of the pandemic, 2019, 2020 or 21. It talks about the generics, and it talks about things that this company did some things well and some things terrible in terms of producing generic. It's not part of this topic, but Ranbaxy did a lot of work on producing drugs from the US to sell, so they very implicated on tech transfer, but they also did a lot of hocus pocus and end up disappearing. But anyway, so this is a screenshot I took some time ago from this company in the US, Patheon, that we work with this company many times, many pharmaceutical industry, that they do tech transfer. They do a lot of things that we can tell them to do, from transfer our technology to scale up our production. So that's as an example. And this is just my glossary. Normally there's so many terminologies, hard to remember. I put this for my students. So this is kind of the end of my two presentations. And if you have any questions, I don't know everything here, but I've been interested to work in the pharmaceutical industry and learn from my colleagues that do a lot of work in the tech transfer. I do more on the chemistry, but kind of very interesting. So anybody has any questions?
Yeah, I see Hayla's question. Hayla's got her hand up. Go ahead, Hayla.
Hi, I have another question. Looking at that tech transfer phase chart that you showed us, do you know what phase a lot of the stuff that we do in the pilot plant falls under?
Let's see. You're looking into I don't work in the pilot plant for the biologics. I work with the small molecules, but
Yeah.
they do a lot of test transfer for different. So you're talking about this slide, am I correct?
Yeah.
Yes,
I'm
we
just-
do. Yeah, in general, we do for the small molecules and the biologics. So for example, the pilot plant at Amgen, they do, they are kind of here, right, in the small batch process. They have to study this to certain level to make sure that when they go from the research laboratory scale, very little, to a relatively a small but relatively large batch process, they can control all the parameters and everything before they can go to the big manufacturing plant. So at Amgen, I visited this before. We do a small batch processes on this, and also I don't know if you have been in building 6 where they do the syringes part. It's very interesting, but it's not easy to enter. You have to have a special permission. They have a big machine. So once they develop in a small batch process a certain amount of the biologic with these big reactors there, big for us, but that's not the biggest for the scale up. They also need to test when the drug gets formulated and then gets into the syringes and it gets this right volume, the right amount, and everything goes well. So we do at Amgen in the biologics a lot of these parts. And then it's when we transfer to a bigger, whether we transfer internally to our manufacturing areas like Rhode Island or Puerto Rico, or whether we transfer to a third party to do it for us. So we're kind of, even in the small molecules, we have, actually it's in building 29, we have our reactors there to do the small batch process, where we study what happened from the laboratory scale to the small, can we produce the right quality of the material when we go from the grams to the kilograms. And this is very important for us because it helps us to transfer the technology, at least at this scale. And then we prepare what we want them to do for the large scales, and comparing, we get the same thing. I hope I'm answering your question. And then the same for us. But this is very critical part. We do a lot of things internally. Many companies do this internally, and then they transfer externally the other parties. Yeah.
Okay, thank you. I was just curious because I've not been to any of the GMP buildings. I'm not sure how their facilities work. The biggest bioreactor we have goes up to 2,000 liters, but I guess I was just curious what part of the process we were in. Thank you.
Yeah.
I have a question.
Yeah, I didn't talk about GMP, but that's another world. Because this is so critical. Anything that goes to humans needs to be highly regulated. The way we work, the way we are educated, we have to make sure that people that do this job has the right skills, the right education, the right expertise, use the right equipment is maintained, control, and the product, the testing, and everything. So that is a huge thing, because we don't want to have something goes wrong and a patients die. That is a no-no. It's a red flag. So yeah, that was a very good question anyway. Yeah.
We have another question by Mohammed.
Hi, it was like a very informative presentation. Thank you so much. And also, let's say we have a situation that like a big company purchase a small company. Would that be considered the technology transfer, or there is like, like some specification in the contracts? And like if that happened and we consider that technology transfer, how long does it take to be correctly done?
That's a very interesting question. Actually, I would say yes. So when, let's say, a big company buys a small company, before it buys the small company, it's going to do a lot of research and a lot of conversations why they are buying it. They're buying it probably because they have some kind of product or technology that's of interest. So the initial, my understanding, I don't do these kind of communications, but the initial communications happens. The small company will not reveal everything to the big company because they don't know they're going to be purchased or not, what's going to happen. There's a lot of back and forth information. And then once this happens and they are agreeable, they actually reveal more the whole thing. So they have to transfer their technology from the small to the big to be able the big to control and to be able to do that product. So yeah, and they have to understand the intellectual property and those kind of things that they might become now in the bigger umbrella of the big company. But so the small company is very careful not to reveal everything because they don't know if maybe the process is going to happen. It maybe fall apart and they don't then. So they when at the beginning on any technology transfer, whether it's a purchase of a company or whether it's a contract, everything is super carefully done with limited information as the conversations goes on and they are more, towards really do it, they kind of start disclosing more things. But there is always very critical with the intellectual property protection. So, overall, if the big company buys the small company to produce that product, they ended have to know the technology. So they have to be a transfer of technology. All this documentation, it has to happen, all these processes, so the big company can continue producing the product or whatever. This is the small company. Many times they even hire the people from the small company because they have the expertise, to be able to do those things. This is very important. So technology transfer happens even internally or even externally because it's the way you do things. You have to tell the others how to make it, so, but good question. So, yeah.
Thank you so much.
We have a question by Howard.
Yeah, let's say like you have a successful anti-cancer drug for animals that you made for animals, and then
Yeah.
you want to use it for humans.
Yeah.
Does it affect how easy your application for the investigative new drug is? Like, would it be easier if it was like successful in animals and you had to go through the same phases for humans?
So yeah, so we do that. So so maybe so basically I'm just gonna basically here in the drug discovery part, we and I kind of put it here. So we do a lot of testing in vitro, in vivo testing on animals. So if the drug is not successful on animals, forget about it. It's not going to go. But so first, for the case of cancer, they also produce animals that have genetic defects. So, or they can have the tumor there. So they have to inject the drug to see if the tumor shrink. And what is if the tumor shrink and disappear, how long it takes, if there's any other effects. So they do a lot of testing. Before we go to phase 1, this has to be really clearly demonstrated that it really cured the animals. Unfortunately, sometimes we cure the animals, but we don't cure the humans. That's a problem and also the type of animals used. A lot of times they do at least 2 types of animals because every type of disease, it has, not all the disease can be tested in the same type of animals. For example, in the case of insulin, pigs are very clearly similar to us. They have similar insulin protein that in humans or things like that. And other times, they'll be using monkeys and rats, or rats and pigs, or rats and dogs and horses. Is kind of nasty, I know. But we have to demonstrate first, I mean, it's a good question, that we can, the disease is really cure or maintain or whatever is the purpose of that disease in animals. Otherwise, those are important proofs. Otherwise, the FDA doesn't let us go forward. So let's say we have a cancer drug for whatever, for the liver or whatever thing, and then we demonstrate in 2 sets of animals that is really that a good job. Side effects is not so high, and whatever, the animals can live freely, whatever, with not the disease. And then we have to prepare a big documentation. And then it's when, if the FDA is convinced that this is really good on animals and in vitro, Then we go to humans and testing and clinical trials. But yeah, this is very important. That's a good question. Very important to test in animal first. We predict in animal the pharmacokinetics and the dose, and we cannot predict what is the human dose. There's a lot of software already used based on experience, based on the data on animals. So what could be the right dose? So maybe from 200 to 500 milligrams, we're going to be testing those doses, even though we never tested on humans, but there is a lot of information in the past with all these drugs that we can predict what is the human dose. And then we test it to see if it makes sense. So yeah, good question.
I guess that's it for questions. Wow, so a lot of information. Obviously, you could spend a year studying this. You could spend a semester. I just wanted to thank Dr. Lipe. Can we give her some appreciation for coming today?
Yeah, and thank you. Fantastic questions, very interesting questions, very exciting. So thank you very much for inviting me. It's always a pleasure to come to your class. It's always super interesting. I think one day I have to take your class. I have to sign as a student because I think it's super exciting, and there will be many, many things that I have to learn myself that I don't know, learning from you.